Introduction. Do you know what is biological classification? Biological classification or scientific classification in biology is a method by which biologists group and categorize organisms by biological type such as genus or species. Modern biological classification has its root in the work of Carolus Linnaeus, who grouped species according to shared physical characteristics. These groupings have since been revised to improve consistency with the Darwinian principle of common descent.
Objectives. At the end of this lesson, you'll be able to define biological classification, study history of classification, identify different kingdoms, understand two kingdoms classification, study three kingdoms classification, know about four kingdoms classification, understand five kingdoms classification, understand six kingdoms classification, understand the characters of kingdom Monera, study structure and characters of Archaebacteria, study structure and characters of Eubacteria. History of classification. Aristotle, 384 to 322 BC, the Greek philosopher, is known as father of zoology. He classified the animals in his book Historia Animalium. Aristotle made two main groups: Anyma, animals with no red blood, invertebrates, and Anyma, animals with red blood, vertebrates. Anyma was further divided into ovipara and vivipara. Theophrastus, 372 to 287 BC, classified plants on the basis of form and texture. He divided them into trees, shrubs, undershrubs, and herbs. Theophrastus gave names and description of 480 plants in his book Historia Plantarum. John Ray, 1627-1705, was first to introduce and define the term animal species. He described more than 18,000 plants and animals in his Storia Generalis Plantarum and developed the key for identification of animals. The modern system of classification began in 1758 when Carolus Linnaeus, 1707 to 1778, published 10th edition of his book Systema Naturae. He also recognized that different species could be grouped into broader categories called taxon based on shared characteristics. System of classification. There are different methods of classification of organisms. According to the number of kingdoms, they are as follows: two-kingdom system, three-kingdom system, four-kingdom system, five-kingdom system, six-kingdom system. Two-kingdom system of classification. Biologists since the time of Aristotle divided the living world into two kingdoms, plants and animals. Carolus Linnaeus also distinguished two kingdoms of living things, Animalia for animals and Plantae for plants. Two-kingdom system was quite reasonable at that period of time since plants and animals could be very clearly distinguished. Three-kingdom system of classification. Ernst Haeckel, a German naturalist, in 1866 created a third kingdom, Protista, to include unicellular protozoa and algae. Four-kingdom system of classification. In the 1930 French marine biologist Edouard Chaton noticed two kinds of organisms, prokaryotes such as bacteria that lack nuclei, and eukaryotes, more complex organisms that have nuclei. In 1938, American biologist Herbert F. Copeland made a fourth kingdom called Monera, now termed Kingdom Prokaryote. Five-kingdom system of classification. In 1969, the American ecologist Robert H. Whittaker proposed that fungi differ enough from other microorganisms to justify calling them a separate kingdom. This led to a five-kingdom scheme of classification comprising Monera, Protista, Fungi, Plantae, and Animalia. Five-kingdom classification is based on complexity of cell, complexity of organism, mode of nutrition, and major ecological role. Whittaker's scheme is still widely accepted as a logical classification of living things. Six-kingdom system of classification. The most recent classification is the six-kingdom system proposed by three microbiologists, Carl Woese, O. Candler, and Emil Wheelis in 1990. Woese, Candler, and Wheelis proposed that all organisms be divided into three major groups called domains above kingdom level: eukarya, bacteria, and archaea. In whose system, the domain eukarya includes four of the six kingdoms: protists, fungi, plants, and animals. The other two domains have just one kingdom each. The domain bacteria contains the kingdom Eubacteria, and the domain archaea contains the kingdom Archaebacteria. Characters of five kingdoms: cell type, cell wall, nuclear membrane, body organization, mode of nutrition. Kingdom Monera. The kingdom Monera includes prokaryote. They lack nuclear membranes. They are also devoid of plastids, mitochondria, and advanced 9+2 strand flagella. Monera cells are microscopic. They are typically unicellular organisms. The predominant mode of nutrition is absorptive, but some groups are photosynthetic or chemosynthetic. Reproduction is primarily asexual by fission or budding. Protosexual phenomenon also occurs. Most organisms bear a rigid cell wall. Example: Anabaena, nostoc, oscillatoria, bacteria. The kingdom Monera or Prokaryota is divided into two subkingdoms: Archaebacteria and Eubacteria. Archaebacteria. Most archaebacteria are autotrophs. Archaebacteria derive the energy they use for their metabolic activities from oxidation of chemical energy sources such as the reduced gaseous ammonia NH3, methane CH4, or hydrogen sulfide H2S, which results in formation of amino acids and proteins. Archaebacteria are divided into three groups: methanogens, thermoacidophiles, halophiles. Methanogens manufacture methane as the result of their metabolic activities. Methanogens die in the presence of oxygen. Thus, they are found in swamps and marshes in which all the oxygen has been consumed by the other organisms that live in these environments. The methane smell characteristics of swamp and marshes is courtesy of the methanogens. Thermoacidophils favor extremely hot and acidic environments such as hot springs. Many thermoacidophils use hydrogen sulfide as their energy source. Halophils survive in very salty environments such as Great Salt Lake and the Dead Sea, which are extremely basic environments. Eubacteria. Prokaryotic cells of bacteria have an outer cell wall that surrounds the plasma membrane, which in turn surrounds a non-compartmentalized cytoplasm dotted with ribosomes. They generally lack membrane-enclosed organelles such as nucleus, chloroplasts, and mitochondria. A circular strand of DNA, usually coiled into one region of the cell, the nucleoid, serves as the single chromosome. Metabolic activities such as electron transport and photosynthesis take place on the plasma membrane, which sometimes folds inwards into the cell's interior. The most common bacterial shapes are rod-like bacilli, spheres called cocci, and the corkscrew-shaped spirula. The cell wall of bacteria contains unique material, peptides. The Gram staining technique distinguishes two types of cell wall construction in bacteria, enabling us to classify them as Gram-positive or Gram-negative. Many bacteria are motile and contain one or more flagella. Eubacteria are of the following 8 types: Actinomyces, chemoautotrophs, cyanobacteria, enterobacteria, gliding and budding bacteria, Pseudomonas, Rickettsias and Chlamydias, spirochetes. Actinomyces are Gram-positive soil bacteria with branching filaments produce spore. Chemoautotrophs are bacteria able to obtain their energy from inorganic chemicals. Cyanobacteria is a form of photosynthetic bacteria common in both marine and freshwater environments, deeply pigmented, cause blooms in polluted waters. Enterobacteria are Gram-negative rod-shaped bacteria, do not form spore, usually aerobic heterotrophs, pathogenic, causing bubonic plague and cholera. Gliding and budding bacteria are Gram-negative, exhibiting gliding mobility. Pseudomonas are Gram-negative heterotrophic rods with polar flagella, very common form of soil bacteria. Some are pathogens of plants. Rickettsias and Chlamydias are small Gram-negative intracellular parasite pathogens of human. Spirochetes are long, coiled-shaped cells with flagella at both ends, common in aquatic environments. They are pathogens. Did you know? Classical taxonomy is based on morphological characters. Phylogenetic taxonomy was introduced by Engler and Prantl. Cladogram is a branching diagram in cladistic which represents the relationship among the species in regard to their derived characters. Carolus Linnaeus coined the words phylum and protista. Red Data Book contains a record of extinct and endangered organisms. Summary. Let us summarize what we have learned. Biological classification of plants and animals was first proposed by Aristotle on the basis of simple morphological characters. Linnaeus later classified all living organisms into 2 kingdoms, Plantae and Animalia. An elaborate 5-kingdom classification was proposed by Whittaker, Mondra, Protista, Fungi, Plantae, and Animalia. The main criteria of the 5-kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationships. In the 5-kingdom classification, bacteria are included in kingdom Monera, and bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity.